All right, let's see. Probably just give another minute. All right, Vance, I think probably now's a good time to start. You want to start by introducing?
Sure, you ready to do this?
Yep.
You don't want to wait longer?
Maybe
just
like
kidding.
another.
I'm just kidding.
If you want to. All right.
Okay, well, thank you everyone for joining us. Today is a really good day, and I'm really excited to introduce Gordon Lau to all of you. Today, Gordon is going to give us his defense talk for his master's thesis. Gordon came to my lab from the MVZ at UC Berkeley, where he was an undergrad and had been working in the lab there with Michelle Koo and others at the MVZ. So I was really excited to get connected with Gordon, and I'm really excited to say that Gordon is one of the first students in my lab to work really closely with AmphibiaWeb and the Amphibian Disease Portal to bring sources of information together and try to get at some bigger questions, not only about where salamanders live and how many there are and all those kinds of basic questions, but also how they may, where they live might affect their ability to survive a pandemic. So, so no further ado, I will just pass this on to Gordon. And Gordon, your title changed a few times, so
yes,
say your title as you start.
Yes.
Oh, and just one last thing. So, so this will go. We're hoping for about a 45-minute talk with questions afterwards, just so that everybody knows if you hang around, there will be the possibility to ask questions afterwards. And finally, I forgot to mention the other two members of Gordon's committee, Michelle Koo and Andy Zink, have both been really instrumental in making, in helping advise Gordon. I really want to thank them as well. So Gordon, with that, please take it away.
All right. Hello, everyone. My name is Gordon Lau, and thank you so much for joining me on my master's defense talk today. And let me just share my screen to my PowerPoint presentation. Okay. Can you all see that? Great. Let's see. Okay. Hi, so my name is Gordon Lau. I am a Master of Science candidate at Vance Friedenberg's lab at San Francisco State University. Joining Vance in my committee, as you already mentioned, is Michelle Koo and Andy Zink. And today I will be presenting on my research, which is the relative influence of life history traits on BD susceptibility in salamanders. All right, so let's start things off with what is an amphibian. So amphibians are a class of vertebrates with over 8,000 species. They're found in all continents except Antarctica. And there are currently 3 extant orders, which are frogs, salamanders, and caecilians. They're characterized by having semi-permeable skin, which allows for water and gas exchange. And as a result, they're prone to desiccation, and as such, they are strongly associated with water. Many species undergo metamorphosis from aquatic gill larvae to terrestrial young adults. However, there are many exceptions, which I will go on in further detail. Right, so why are amphibians important? They provide a number of ecosystem services, such as they help keep arthropod populations in check. They themselves serve as an important food source for many animals. They help the carbon cycle by regulating the movement of carbon through the food web. And additionally, they serve as indicator species, which means that they signal the health of the environment. And that said, unfortunately today, several species of amphibians are threatened worldwide, or as I like to call it, the plight of the amphibians. And so what is the plight of the amphibians? So right now we're witnessing a global amphibian decline. Scientists estimate that about at least 1/3 of all amphibian species are threatened with extinction, if not more. And there are many reasons for this, such as pollution, habitat destruction, and a big one is disease. And so perhaps the most destructive disease in amphibians is that of Batrachochytrium dendrobatidis, or BD for short. And this is a fungal pathogen that is implicated in amphibian decline, and it causes a disease known as chytridiomycosis. And recently, a second related fungus was discovered called Batrachochytrium salamandrivorans, or B-cell for short. It infects salamanders primarily. However, it may also infect frogs and caecilians. And here's a picture showing the life cycle of BD. So first thing what happens is the BD zoospores, they burrow into the skin of the amphibian. And then over time, the BD zoospores mature into this fruiting body called sporangium. Eventually, the sporangium will release spores into the environment. And if the spores are released into the water, what the zoospores will do is they will swim in the water using flagella. And if they contact other amphibians, they will infect those amphibians. The cycle repeats. Alternatively, another mode of transmission is one infected amphibian touching another amphibian. So having direct skin-to-skin contact between amphibians is also another way of transmitting the fungus. And so what are the effects of BD chytridiomycosis? So first, you might see skin hyperkeratosis or thickening of the skin, and this leads to osmoregulatory stress. In many cases, you see lethargy, loss of appetite, skin sloughing, which is where the skin sheds in sheets and layers, and in several cases, death. Where do BD-related declines occur? And so here's a map from Fisher and Garner 2020, just giving you an idea of where those BD-related declines are happening worldwide. And I do want to clarify that the declines that we're seeing are primarily due to the worldwide invasion by one lineage of BD, which is called BD-GPL, short for the global panzootic lineage. That likely originated from Asia and was presumably spread throughout the rest of the world as a result of the global human trade. And so we see that declines are prominent in areas without endemic non-pathogenic lineages. So you see in this map that BD occurs in many places worldwide. And besides BD-GPL, there are other lineages of BD that are endemic to certain areas. So you see there's BD Asia 1, BD Kate, BD Asia 3, BD Asia 2/Brazil. And so there's many regions in the world that have these endemic lineages of BD. And so in these places, the amphibians have presumably coexisted with them for a very long time and have evolved a resistance to BD, such that when BD-GPL has invaded their area within the past several decades, that since they've already built up a resistance to their own native lineage of BD, they are much more resistant to this new introduced lineage of BD, BD-GPL. That said, we do see a lot of declines in areas that do not have their own native endemic lineages of BD. And so these hotspots include the Western United States, Mexico, Central America, South America, Africa, and Eastern Australia. And I want to bring attention to the salamander decline in North America. And so right now, so there's a high salamander biodiversity and a lot of associated declines with that in BD novel regions, such as the Western United States, Mexico, and Central America. And you can see here in the species richness map that there are a lot of salamanders in the Western United States, and there's some zones of red, which indicates high species richness in Mexico, as well as in Central America. And you will also notice that the Eastern United States is very red. However, I am only focusing on the Western United States, Mexico, and Central America, because these are places that are novel or naive to the pathogen, whereas BD has existed in the Eastern United States for a longer time, and we don't see die-offs associated with the Eastern United States. These declines are also coincident with the invasion of BD. So if you look at the graph on the right from Chang et al. 2011, you see that over the years, the abundance of Mexican salamanders has declined drastically as the prevalence of BD has increased in the environment. And on the figure on the left from Lips 2006, you will see that you will see the rate of expansion of the BD fungus invading throughout Central America over starting from the late 80s all the way through the early 2000s. And you will notice that there's been a lot of expansion of the fungus, especially through the 90s, indicating that this pathogen is very recent and it's wreaking a lot of havoc. And I do also just want to clarify, I'm emphasizing the word coincident because we still don't have enough strong evidence to say definitively that BD invasions are directly responsible for the decline in salamanders. However, the two do coincide. So there is reason to suspect that one affects the other. Right. That said, all salamanders may be infected, but some are more infected than others. So not all salamanders experience BD-related declines in these BD epidemic regions. And so it's possible that maybe these species exhibit BD-susceptible traits. And so one example of that is the study from Weinstein 2009, looking at Batrachoceps attenuatus, also known as the California slender salamander. And what she did was she housed these slender salamanders in two different treatments. So one treatment was in wet housing conditions and the other was in dry housing conditions. And then she inoculated salamanders in both treatments with BD fungus, and she recorded survivorship over time. And as you will see over the course of her study, all the salamanders in the dry conditions survived infection, whereas only 40% of the salamanders in the wet treatment survived infection. So what this suggests is that, for one thing, we know for a fact that the California slender salamander is indeed susceptible to the effects of chytridiomycosis. It can indeed die from it. However, if put in an environment that is less suitable for BD fungus, as you may recall, that BD fungus likes wet environments. So if you have these salamanders in dry conditions, they could potentially put themselves in a microclimate that is less suitable for BD growth, and if they have traits that can help accentuate the fact that they are in these dry conditions, it could potentially help them shed themselves of the infection. And so this brings me to my main question, which is, what is the relative influence of salamander life history traits on BD infection susceptibility? And so a case study looking at why BD and traits might be related, why they might be important, is that of the Queensland tropical rainforest frogs from a study by Riley and Alfred 2007. And so if you may recall that Eastern Australia is one of the places that has been hit really hard by BD. There's a lot of amphibian declines of that area, of which Queensland is an example. And so in this study, the scientists looked at 3 sympatric species of frog in an area that was hit hard by BD. So they first looked at Latoria nonotus. This is an endangered species, has long-lasting population decline. They looked at Latoria genomaculata, which is a least concern species, originally had population decline, then recovered since. And lastly, Latoria lusuri, which is a least concern species, has not witnessed any population decline. And so what they found was, when looking at different types of traits, that nonotus had by far the highest amount of stream contact at 84%, compared to 16 and 13% for the other 2 species. And when looking at frog-frog contact, nonotus also had by far the highest frequency at 28% versus 3% and 2%. Now, what this suggests is that all 3 of these species, even though they are vulnerable to the effects of BD infection, they're vulnerable to getting infected, nonotus is the one that is endangered because it engages in stream contact the most and frog-frog contact the most, which are the 2 main modes of BD transmission, thereby putting it at an elevated risk of coming into contact with BD and contracting the infections from it. And so you're probably wondering, why include a study in frogs if I'm talking about salamanders? So the reality is, frogs are much more easy to study than salamanders. They're much more easily observable. They're out in the open. It's much more easy to find them, and they also, vocalize versus salamanders. And there's also just more frog data available. That said, even though a lot of these types of studies do focus primarily on frogs, it's still worth looking at these studies just by virtue of the fact that, both frogs and salamanders are amphibians and they do have shared physiology. So it's still worth looking at these frog studies and see if we can find similar trends in trait-related studies for salamanders. And another case looking at BD and traits is that of Greenberg et al. 2017. This was a lit review study showing that some amphibian traits predict BD infection prevalence. And so the scientists found that there's higher BD prevalence in species that are more aquatic, and also species that had more rapid sexual maturation of under a year. And even though the study includes 302 species from all amphibian orders, salamanders themselves may help contribute to the trends that we see in traits and infection. And so yes, again, so why study salamander traits in BD? So there's less traits and trait diversity to focus on than frogs that we know of, so it makes for a slightly more simple study. And there's less species than frogs, so it's logistically less demanding to do such a type of study. There's only about 700 something species of salamander versus well over 7,000 species of frogs, so it's at least a tenfold difference. And most salamanders, which are in the family Plephodon today, are actually non-aquatic. So this is a good case study to look at the role of traits with a pathogen that is primarily aquatic. All right, so right, so now that we're talking about traits and BD susceptibility, so what types of traits are these? So again, as you may recall, BD may transmit through contact with infected bodies of water and substrates. Salamanders may also vary in preference for aquatic life stage from the larval, juvenile, and adult stages. You see a lot of heterogeneity in preference for aquatic life stage. Some species of salamander stay in the aquatic gill larval form their entire lives. Some have the aquatic gill form as larvae metamorphose into young adults. And then yet other species are terrestrial for their entire lives, from the moment they hatch to the moment they become adults. And so variation in these types of traits may result in varying levels of BD contact. Right, then what about other types of BD susceptible traits? BD, again, may also transmit through skin-to-skin contact between individuals. And so salamanders vary in degree of intraspecific contact, which deals with social behaviors such as territoriality, cannibalism, group breeding, communal nesting, and parental care, among other traits. And again, variation in these types of traits may also result in varying levels of BD contact. Right, so now moving on to materials and methods. So first things first is to identify our areas of interest. Again, that is the Western United States, which I define as west of the Rockies, Mexico, and Central America. And the reason I chose west of the Rockies to define where the Western United States is it acts as a good natural boundary for where the Western US is, as salamander biodiversity is very low in the central US, just east of the Rockies. So if you remember from that map that I showed earlier of salamander species richness, you see a lot of red on the west coast of the United States, and then you see a lot of red on the eastern part of the United States. But the central part is not especially specious for salamanders. So the Rockies act as a good natural boundary for that. And so yeah, again, why not include the eastern US east of the Rockies? It's because BD is an endemic pathogen in the eastern US, and studies show that there's a lot of BD persistence in that area, but no related BD die-offs. And so an inclusion of eastern US salamanders, despite a high biodiversity, would actually confound a study on traits and BD susceptibility, just because they're not known to die from BD. Okay, and so here are my three regions. And you can see I've drawn a black line approximating where the boundary for the Rockies is. So we have the Western United States, Mexico, and Central America. All right, and then from there, it's the species list compilation. So I compiled a list of salamander species from the Western United States, west of the Rockies. Also compiled species from Mexico, Belize, Guatemala, Honduras, El Salvador, Nicaragua, Costa Rica, and Panama. And ultimately, I recorded 352 species in 6 families: Embystomatidae, Dicamptodontidae, Plethodontidae, Rhycotritonidae, Salamandridae, and Sirenidae. And unfortunately, 2 island species I had to drop from the study. They were both in the genus Oedipina, which is a plethodonid, due to data limitations, which I will explain later on in the method section. So ultimately, I only had 350 species in my analysis. And just to give you an idea what the, continuing on, Plethodontidae is primarily a non-aquatic family, like I mentioned earlier, whereas all the other families in my study, all of them have aquatic life stages. And that is important to keep in mind. And here are this. Okay, another thing is Plethodontidae makes up the majority of the species. Out of my 350 species, 316 of them are plethodonids, or 90.3%. And so here are the different families in Embystomatidae, which are the mole salamanders, the Camptodontidae, which are the Pacific giant salamanders, Plethodontidae, the lungless salamanders, Rhycotritonidae, the torrent salamanders, Salamandridae, which includes the group of salamanders called newts, and Sirenidae, which are the siren salamanders. Right. And so from there, I compiled life history trait data sets. I downloaded and organized these life history trait matrices for my 350 salamander species from AmphibiaWeb, as Vance mentioned, and it is an online database for amphibian information. And there were missing values in the data set, so I would fill in the missing data with literature searches whenever possible. And ultimately, I identified 13 traits that may be relevant to BD infection susceptibility and show sufficient variation to conduct comparative analyses. And so I split these traits by relevant to BD transmission. So traits relevant to water contact, as well as intraspecific contact. And so here are the traits to water contact, relevant to water contact. I'll go through them briefly. So you have general adult habitat, which can be aquatic, non-aquatic, or both. Aquatic adult habitat, which is either lotic or lentic. Lotic being fast-flowing waters like streams, rivers, and creeks. Lentic being waters that are stagnant still, such as ponds, pools, lakes. And then I looked at non-aquatic adult habitat. Is it subterranean, arboreal, or terrestrial? Meaning location, aquatic, non-aquatic, or both. Egg deposition site type. Is it aquatic or non-aquatic? egg deposition aquatic site type, again, lotic or lentic, or phytotelma, which is basically plants that have a cup-shaped cavity that allows them to collect water. And some species amphibians do indeed lay their eggs in these phytotelma. And an example of this are the bromeliads. Egg deposition aquatic site water permanence. Is the water permanent or is it ephemeral? Egg deposition non-aquatic site type. Again, do they lay their eggs in subterranean or arboreal or terrestrial habitats? And larval aquatic habitat, lotic, lentic, or phytotelma. Then here are the traits that are relevant to intraspecific contact, territoriality presence, immunonesting presence, egg guarding behavior, and cannibalism in larvae. Okay, so modeling BD exposure risk. So unfortunately, food infection data of BD is not available for all species of salamander. So in order to get around that, I generated a habitat suitability model, which I'll abbreviate HSM, using GIS methods or geographic information system methods in order to model the likelihood of salamander species contacting high BD suitability areas in its range. And I took many steps to minimize overfitting of the model, which I will get to in a second. And this relative risk of salamander species exposure to BD in the field, as per the HSM, was used to run my trait/BD correlation analyses. Right, so BD sites. So I compiled occurrence data sets of BD positive samples, of which I had 962, and BD negative samples, of which I had 9,732, from my relevant regions from amphibiandisease.org or directly from the papers, or sometimes I would even contact the authors of the papers to get the data sets. And so here are the papers that I use if you're curious. And so here are all my points plotted out. So BD positive occurrences are in red, BD negative occurrences are in gray. So you see that there's quite a clustering in California, as well as in southern Mexico and Central America. And I will address this in a little bit. And then the next thing is to select bioclimatic variables. And so I use bioclimatic variables in order to inform the suitable habitat for a species of interest, which is BD, based on temperature and precipitation. And so I downloaded 19 bioclimatic variable raster layers from WorldClim in 2.5-minute resolution. And so these bioclimatic variables are basically different variables relating to temperature and precipitation that are deemed biologically relevant. And then I use the R function extract in the package raster to extract bioclimatic values from my occurrence points. I generated this into a matrix. Then I use the R function core in the package stats, and I calculated the Spearman's rank correlations to determine highly correlated variable pairs from the point value matrix. And the reason I'm doing this is because a lot of these bioclimatic variables may be highly correlated. And if you have a lot of highly correlated variables, you risk overfitting of your model. And you don't want an overfitting of the model because that doesn't make for a good model. So this is why I ran these rank correlation analyses. And so for my variable pairs, when R squared was greater than 0.7, I kept the higher number of variables within the variable pair. And so this resulted in a subset of the variables bio 7, 10, 11, 15, 16, 18, and 19. I just go through them briefly. So 7 is temperature of annual range. You have mean temperature of warmest quarter, mean temperature of coldest quarter, and the other 4 relate to precipitation. So precipitation of seasonality, precipitation of wettest quarter, precipitation of warmest quarter, and precipitation of the coldest quarter. Right? And the next step is the bias file creation. And so again, I need to account for the presence of these negative sites as well as the clustered uneven sampling. And so creating what they call a bias file basically helps direct model training to these, what they call pseudo-absence areas, to better predict suitable areas. And so I use the R function rasterize in the package raster to rasterize my negative point vector layer with bioclimatic raster layer values. And then I use the R function KDE2D in the package mass to estimate the 2-dimensional kernel density of my negative sites, and I exported this as my bias file. Right. Determining model settings. And so the biggest issue of HSMs, again, is overfitting, which is when the function is too closely fitted to our limited set of data points. And so in order to account for this, I ran the R function enm evaluate and the package enm eval to identify which model settings I should use that balance model fit and predictive ability. And what it does is it returns a matrix of model settings and corresponding delta AICC values. Now, these delta AICC values, what they basically tell you is it's a comparison of different model parameters, different model settings, and the settings with the lowest delta AICC values are the ones that you want to use for your model. And so the model settings that you can choose from are the regularization multiplier, RM for short. This limits the model complexity and generates a less localized prediction, as well as feature classes, which are basically different features that help, are different ways of mathematically calculating how to generate the model. So there's a linear, quadratic, product, threshold, and hinge classes. And so I chose the following settings based on the lowest delta AICC from E&M Evaluate. I had a regularization multiplier of 1, and I used linear, quadratic, product, and hinge features. So now running everything in this program called MaxEnt. So I developed a BD habitat suitability model using MaxEnt 3.4.1 modeling software. Now what MaxEnt is it's a software that uses occurrence points for your species of interest, and it uses environmental layers such as the bioclimatic variables that I was talking about to generate a map suitability. In order to constrain the background area used for training, I used QGIS to create a minimum convex hole around my points. And then I use that to clip my subsetted bioclimatic variables to input into MaxEnt. And then from there, I input the BD positive records, I input the bias file, and I set the settings to RM equals 1 and linear, quadratic, product, and hinge features. I use the default of 10,000 background points to train my model. Continuing on, I randomly assign 20% of my presence points to calculate the area under curve graph to assess my model fit, which I'll explain later. I also use the minimum training presence threshold to inform areas of general BD suitability. Now, what the minimum training presence threshold basically tells you is that below a certain suitability level, that is considered when BD cannot grow in that place at all. So if it's underneath a certain level, then suitability just straight up drops to 0. And so that's what the threshold level is saying. And then I ran a k-fold cross-validation using 50 k-fold replicates. And then the results of these k-folds were all averaged together to create the final habitat suitability model, which I exported in C-log output format. And then I projected this to a map of North America. And so now I need to assess my model, make sure the model fit is good. Yeah, so I just evaluated the model to make sure it was a good predictor of BD suitability. And so I looked up the area under curve graph, or the AUC graph, and what this graph is it describes the relationship between the proportion of observed presences that were correctly predicted and the proportion of observed absences incorrectly predicted. When AUC is 1, the model perfectly predicts presence from absence. When AUC is 0.5, the model prediction is no better than random. And so the model that I generated had a mean AUC of 0.975, therefore it predicts presence from absence very well. Additionally, I checked the response curves to make sure they make biological sense. So response curves are these graphs that MaxEnt generates that compares suitability versus different bioclimatic values for each variable. And so when looking at these response curves, basically you just want to see that they fall reasonably within the species biology. And so BD has optimal growing conditions at 17 to 25 degrees Celsius and cannot survive above 30 degrees Celsius. So I verified this by looking at the single variable response curves for Bio10, which is the mean temperature of the warmest quarter, and Bio11, which is the mean temperature of the coldest quarter. So let's look at Bio10. You see that peak suitability is around 20 degrees Celsius, which falls in line with the 17 to 25 degrees Celsius thermal optimum. And then by the time it reaches 30 degrees Celsius, suitability drops pretty much to 0. Whereas in Bio11, the trend is not nearly as clean. You do see a peak in suitability around just before 10 degrees Celsius. And then again, it drops off to 0 suitability by the time it reaches 30 degrees Celsius. Now, how I interpret this is even though the peak of around 10 degrees Celsius for suitability doesn't fall in line with the 17 to 25 degrees Celsius, one way to think about it, since we're looking at mean temperature of the coldest quarter, potentially speaking, it could be that when you're in when you're in the cold part of the year, any spikes in temperature could impart some thermal stress on the fungus. So that's just how I see it. But overall, the model fit looks fairly good. Okay, and so here's the habitat suitability model. Again, I have the Rockies drawn out, the boundary for the Rockies drawn out. You can see the positive and negative BD sites. And then here you can see the colors for low suitability versus high suitability. And here's the same map without those points. Right. And after we have that comes the salamander range maps. So I downloaded species range maps from AmphibiaWeb, as well as one map from IUCN when that map was not available from AmphibiaWeb. And I visualized this using QGIS. So here are all the salamander range maps. You will notice that there are two species where their ranges fall quite out into the eastern United States, Embystomum mavortium and Siren intermedia. The reason I included them in the study still was because for Embystomum mavortium, a good chunk of its range does occur in the western United States. And for Siren intermedia, even though most of its range is in the eastern United States, there is a small part of it that actually occurs in Mexico. So for those reasons, they fell into my regions of interest, and I included them into my study. And so here's just kind of a close-up of the western United States. You can see there's a clustering of species, especially around California, here in Mexico and Central America. You can see that actually a lot of these species have very small ranges. And then now comes the infection index calculation. In the absence of infection susceptibility data for several of my species, I use what is called the infection index, or basically it's the likelihood of a salamander species contacting high BD suitability areas in its range. And so I got those range maps and I overlaid them onto the BD habitat suitability model. And then using zonal statistics and QGIS, I calculated the average suitability from 0 to 1 of a HSM that was captured within each salamander range map polygon. And then going back to those two species that I dropped, so I dropped those two species of Oedipina from the study because since they lived in islands, unfortunately, their ranges did not intersect the habitat suitability model of BD, so I was unable to gather any information from them. So here's what that looks like. You have this range maps overlaid on the habitat suitability model. And just to give you an example of what is actually going on in terms of those calculations, so here are two example species, Dicamptodon copi and Plephodon stormi. So the idea is you take the range map of the species, you overlay it on the habitat suitability model, and what that does is it takes a cookie cutter shape of the habitat suitability model, it captures all the pixels within the habitat suitability model map, and each pixel has a value in it from 0 to 1. Then what zonal statistics does is it calculates the average value of all those pixels within the range map, and that average value is what I consider to be the infection index. So for Dicamptodon copi, its infection index is 0.38, which means it's sort of in moderate BD suitability habitat, whereas Plephodon stormi has an infection index of 0.9, which means that it occurs, much of its range is in very high BD suitability habitat. Okay, then now comes the next part, phylogenetic analysis. So I need to determine how much evolutionary history affects traits in order to determine if the infection risk is purely trait-borne or has a genetic component. So I downloaded and organized the amphibian phylogenetic tree from the paper of Yetson Pyre in 2018 using the R packages, ape, Geiger, and phi tools. Then I calculated Pagel's lambda for each trait using the R function phylosig and the package phi tools. A method that was also used in Greenberg 2017. Now what Pagel's lambda calculates is it looks at how closely does the evolution of traits in a certain taxonomic group, how closely does that follow the rate of evolution within the group itself. So when you have a lambda of 0, that means that there is no phylogenetic signal in the traits. Therefore, trait evolution is not closely associated with the evolution of the salamanders, whereas a lambda of 1 means you have strong phylogenetic signal, where trait evolution is closely associated with the evolution of the salamanders themselves. Then I also calculated the p-value for the Pagel's lambda test just to see how significant this test was. So when p is less than 0.05, that means that the results of the test are significant, and then p over 0.05 means that the test is not significant. And then next part, boosted regression tree analysis. This is a machine learning technique described in Elif et al. 2008. And what it does is it builds a prediction model by combining multiple binary decision trees from the data set. And it basically predicts which predictor variables, which are the traits in this case, affect the response variable the most, which in this case is the infection index. And so the model stops generating new trees when the minimum predictive deviance is reached, or when each tree generated by the model differs from the next tree by the least amount possible. And the reason it stops is because having too many trees leads to overfitting, which is an issue I've already described. And so here's an example of what these regression trees look like. So basically you have a sample of individuals and you split them up by certain traits. So in this example, you can split them up by age, you can split them up by gender, You can split them up by their computer usage. And basically what the model does, it will keep splitting up for each tree, it'll keep splitting up the group until the groups can't be split up any longer, or you've used up all the traits, or you've set a certain what they call a tree complexity, which is the amount of split points or the amount of traits you want to include in each tree to split up your group. And so once one of those is reached, that tree stops, and then it generates a new tree based off of existing knowledge of the prior tree before that. So pros over GLMs, or generalized linear models, which are a commonly used regression analysis technique. And so one of the pros is it doesn't assume a linear structure to the data. It assumes that the data is nonlinear, and it builds a model to fit the data instead of vice versa with GLMs. Another pro is it's robust to missing data. In my trait data set, even though I tried my best to fill out a lot of the values, there were still a lot of other missing values from my data set. And so what's nice about boosted regression trees, BRTs, is they help, they are able to work despite the fact that you have missing data. And so in terms of how I actually ran things, so I performed my BRT analysis using the R function gbm.step and the package gbm, and I did this for all species by family and by genus. And I set the infection index as my response variable, and I set the traits as the predictor variables. I removed traits that lacked data or interspecific variation for each of my BRT model iterations for my different taxonomic groups. Then I would record what is called the relative influence values of traits. So every time the model finishes running, it returns a list of the traits that are important to infection index, and also returns a number called the RI value. It's a value from 0 to 100, which indicates the percent influence that that variable has on the infection index. So a value of 90 means that it contributes a lot to the model, for example. Right, and so here are the boosted regression tree parameters. If you're just curious, I'm just going to go. You don't have to read too much into this, but just look at the top graph on the left. So here you see species included, a number of species, learning rate, which is how much does each tree contribute to the final overall model. That's one way you can see it. The tree complexity, which I described as the number of split points you want to have in your tree, how complex you want your tree to be, how big you want your tree to be. Bag fraction, which is basically the random sample of it basically refers to what proportion of your sample is to draw from randomly each time you generate a new tree. Number of trees, which is just the output that you record at the end. It tells you how many trees were needed to generate the model. And then the minimum predictive deviance is the value indicating how much each tree differs from the next. And the minimum value, once it's reached, and once predictive deviance goes up, then the model stops and it tells you what the minimum predictive deviance is. And so in terms of criteria for these parameters, so I set, for the boosted regression trees, I set the error distribution to normal or Gaussian distribution. I set the learning rate to no greater than 0.001, and occasionally I would decrease that if needed for the model. The bag fraction I would always set to at least 0.5, sometimes greater if the model said that there were not enough samples to run in my model. So basically, by increasing it to higher numbers, you're essentially, if you have a small sample size, like only like 5 or 10 species, if you set it to like 1 or 1.5 or 2, you're essentially duplicating the number of samples you have, and that helps the BRT analysis run more smoothly. And another thing is that the number of trees always has to be at least 1,000. This is a number recommended by ELIF 2008, telling you that if you have at least this many number of trees, then you can be reasonably sure that your model is good. Right. And then after that, you put everything together to look at the relative role of salamander life history traits to BD susceptibility. And so the first thing is that there should be significant trait relative influence value disparity. So if you think about it this way, so there's 13 total traits. And so if a prediction of which traits are influential is no better than random, that means that each trait contributes only roughly 7.7 RI to the final model. 7.7 times 13 is 100. So if you get your output of traits and all of them have similar RI values, then that just means that the model predicts things no better than random. So you do want to see significant disparity to actually confidently say that one trait is actually more important, is actually important, contributes to some meaningful degree to the outcome of the model. The RI value should be high enough. I designated this as at least 10. Now, the reason I chose 10 as my cutoff threshold number is because, again, it's like a nice even number that's a little bit above the 7.7 that I was describing just earlier. And also when I ran the BRT analyses, I saw that there were multiple instances where a majority of the traits actually had very low RI values under 10. So I figured that 10 would just be a nice cutoff to say which traits are actually important to BD susceptibility, which traits are not really that important to BD susceptibility. And then last part is to determine if the traits have phylogenetic signal. And so here is a table showing the different categorizations of the traits as based on their Pagel's lambda values. So let's just focus on the, the top three, the green, yellow, and red. So again, all of them should have, all the traits should have an RI of at least 10. And then Pagel's lambda of 0 to 0.3 means it has a low phylogenetic signal, which I colored green. A Pagel's lambda of 0.3 to 0.7 has moderate phylogenetic signal, which I color-coded yellow. And 0.7 to 0.1 for Pagel's lambda means it has a high phylogenetic signal, which I color-coded red. And you also notice that I included the p-values for these. So ideally, you would want to have p-values that are less than 0.05, which indicates that the Pagel's lambda test is high significance. And then for the bottom three traits, the Pagel's lambdas are the same, so low, moderate, and high phylogenetic signal. However, because their p-values are greater than 0.05, we can't reject the null hypothesis. Therefore, the Pagel's lambda values are not as informative as they could be, and hence I have color-coded them gray. All right, so now moving on to results. And so when looking at all species, I found that there were 4 traits that were important. So I'm just going to name them off. So non-aquatic adult habitat at 36 RI, non-aquatic egg deposition site type at 31. These 2 are water-based traits. And the bottom 2 are the intraspecific contact-based traits. So we have egg guarding behavior at 15 RI and communal nesting presence at 12 RI. You notice that they all have really high Pagel's lambda values at over 0.7, and all of them had p-values of less than 0.05, which means that all the Pagel's lambda tests for these 4 traits are significant. And then here on the right, you can see the number of observations, which just basically means how many species did I have in this particular run of the BRT that had a trait value, had a value associated with that trait. Right? And so here are the graphs for high RI traits that are relevant to water contact for all species. So you can see on the top is non-aquatic adult habitat, the bottom is non-aquatic egg deposition site type. And I've abbreviated arboreal, subterranean, terrestrial as A, S, and T. And some of them have habitat preferences or egg deposition site preferences that is a combination of at least 2 or even 3, all 3 of these habitat types. So you notice that for the top graph, that species that are terrestrial and subterranean have the highest average infection index compared to species with other types of habitat preferences. And generally speaking, you notice that species that display affinities for an arboreal habitat generally have a much lower infection index. And then looking at the bottom graph for non-aquatic egg deposition site type, again, you see that species that deposit their eggs in terrestrial and subterranean environments have by far the highest infection index. Trends are somewhat similar to the non-aquatic adult habitat. However, you will notice that for species that are arboreal, terrestrial, and subterranean, egg depositors, so all 3, have the second highest infection index, which is different from what we see in non-aquatic adult habitat. However, I do want to caution into reading too much into this because for that trait value, we only had 1 species that in my study that laid their eggs in both or in arboreal, terrestrial, and subterranean environments. And so there's only 1 species having that value. So you would obviously want a larger sample size to say something more meaningful about, about that value. So just keep that in mind. And then when looking at the high RI value traits relevant to intraspecific contact for all species, we see that in terms of egg guarding behavior, species that do egg guard have generally higher infection index than those who don't. However, the spread is still pretty wide. That I would caution against reading too much into it. And then meanwhile, for communal nesting presence, you will see that species that communally nest have a much higher average infection index than species that don't communally nest. And just to note that much of these trends that we see and communal nesting presence are driven primarily by Batrachoceps. So the slender salamanders, they're one of the very few genera that do communally nest. And so they are the ones that are driving most of this difference that we see in this trait value. And also just another note, you will recall that plethodontids make up 90% of my species in my study. And so it's reasonable to assume that a lot of the trends that we're seeing in these high RI value traits are due primarily to Plethodontidae. So from there, I went to look at each of the families on their own and see if we can find any other differences there. So just looking again at Plethodontidae by itself, you see again, it's the same four traits that come up in the same hierarchy. So non-aquatic adult habitat at 38, non-aquatic egg deposition site type at 32, egg guarding behavior at 15, and communal nesting presence at 14. And you notice that non-aquatic egg deposition site type has a Pagel's lambda of less than 0.1, and it's also significant, which is why I color-coded it green. Whereas the other three traits, all of them had high Pagel's lambda values and were significant, meaning that unlike the former trait that has a low phylogenetic signal, these three other traits have very high phylogenetic signals. Okay, and then again, looking at the high RI value traits for water contact, the trends are pretty much the same, so I'm not going to get into them. They look pretty much the same as when looking at all species. And again, the same thing when looking at high RI value traits relevant to interspecific contact. The trends are pretty much the same as for all species, which makes sense again, as they make up the vast majority of the species in my study. So I wanted to look at some of the other families to see if there are other trends in them. So I looked at next was Embystomatidae. And what you see here is you have two traits that are important. So you have the aquatic adult habitat with an RI of 87 and general adult habitat with an RI of 13. And even though both of them had a Pagel's lambda of less than 0.1, unfortunately, both their p-values are greater than 0.05. Therefore, we cannot reject the null hypothesis about Pagel's lambda tests in terms of what the phylogenetic signal is. And so you would see here is the graphs for high RA values for water contact-based traits. And you see that for adult aquatic habitat, species that are lotic have by far a much higher infection index value than species that display any affinity for lentic waters. And then when looking at general adult habitat, you see that species that are exclusively aquatic have generally higher, somewhat higher infection index than species that are non-aquatic/aquatic. So species that display any affinity for a non-aquatic lifestyle. When looking at Salamandridae, we see that territoriality presence, which is actually a social behavior, has the highest RI at 83. Aquatic adult habitat comes in second at 14. And even though Pagel's lambda was less than 0.1 for both, again, their p-values were high at 1, so it's greater than 0.05. Therefore, you cannot reject the null hypothesis. And therefore, this value you should take with a grain of salt. Although, again, since we're only looking at 5 species, to have a greater p-value. And so here is the water contact-based trait association for salamandrids. Looking at aquatic adult habitat, you see that on average, species that are exclusively lotic have a higher average infection index than species that are both lentic and lotic, so species that display any affinity for lentic waters. You'll see here that regarding territoriality presence, that species that are territorial have a higher average infection index than species that aren't. However, due to the widespread in infection index values for non-territorial species, I wouldn't read too much into this graph. And unfortunately, I was not able to generate any BRTs for Dicamptodontidae, Rhycotritonidae, or Sirenidae due to the low diversity of species as well as low diversity of trait values. And then when I ran the BRT analyses by genus, I saw that pretty much showed similar trends in traits and average infection index values to my family analyses. And so you see that the Plethodonta genera mostly have traits that are relating to non-aquatic adult habitat / egg deposition site. We see that, meanwhile, Batrachoceps had high RI values for social behaviors. And so the two social behaviors that showed up in the Batrachoceps BRT were communal nesting presence and egg guarding behavior. Communal nesting presence, which makes sense because Batrachoceps is one of the very few genera that communally nest. And then when looking at Tarica, a genus in Salamandridae, it has one high RI value trait, which is aquatic egg deposition site type. And then all these high RI value traits either had moderate to high phylogenetic signal, which was either both significant or non-significant, or they had low phylogenetic signal that was non-significant, meaning that for the most part, there were no BRT model iterations that showed low phylogenetic signal that was significant. And so a trait value summary for plethodontids, you see that species that are both terrestrial and subterranean have a higher infection index than arboreal species. And then we see the same trends in egg deposition site. You see the communonesters have higher infection index than non-communonesters, and egg garters have higher infection index values than non-egg garters. However, the index values are very spread out. Meanwhile, for Embystomatidae and Salamandridae, we see that strictly aquatic species have slightly higher infection index than non-strictly aquatic species. We see that lotic exclusive species have higher infection index than lentic species. We see the same trends in egg deposition site type. And we see that territorial species have higher infection index than non-territorial species. However, again, the infection index values are highly spread out. All right, so coming from that, so now we can identify which species are at risk. And so these are species with high infection indices and display trait values that are ideal for BD infection risk. And so at-risk species, I define them as having an infection index of at least 0.5 and are both terrestrial and subterranean if non-aquatic, or they live in lotic waters if they are aquatic. And so here are the breakdown of those species. So when looking at Plethodontidae, you see Batrachoceps with 19 out of 22 species, Hydromantes 5 out of 5, Oedipina 2 out of 36, Plethodon 1 out of 9, Pseudoeurysia 1 out of 39. Looking at Rhyacotritonidae, for the genus Rhyacotriton is 1 out of 4. For Salamandridae, you see 1 out of 4 species, and Pterica is at risk. For Embystomatidae, you see in the genus Embystoma, 3 out of 20 species are considered at risk. Right, so now moving on to discussion. And so looking at the trait differences between aquatic and non-aquatic families, we see that high RI traits relating to habitat type suggest that contact with water is important to BD infection risk in both non-aquatic families, which is the plethodontids, and aquatic families, which is all other families in my study. And so this supports previous studies relating to BD and habitat use, such as the Raleigh paper looking at, again, the Queensland frogs and seeing that frogs that were that contacted streams more or at higher risk of BD infections, as well as this other study in Cameroon looking at frogs in Cameroon. And what they found was that frogs that were arboreal had lower infection indices than related species that were closer to the ground and utilized streams and ponds more. Then a study to measure the frequency of water source contact would help improve the BRT results. And when looking at the specific habitat effects, the habitat trait values on BD infection susceptibility, we see that for non-aquatic species, species that are both terrestrial and subterranean have the highest infection index, whereas arboreal species generally have the lowest infection index. This could be because that the terrestrial and subterranean habitats are closer to water, which is, where BD likes to live. It could be that the ground temperature fluctuates less than air temperature, since ground acts as a good buffer against shifting temperatures, fluctuating temperatures, versus if you're in the treetops, you're more subject to the fluctuations in air temperature. And having less fluctuations in air temperature could be better for BD growth. Meanwhile, for aquatic species, lytic species that have the highest infection index, lytic species have the highest infection index, whereas lentic species have the lowest infection index. Could this be because lytic species are exposed to more water and potential spores? But if you think about it, wouldn't it make sense that lentic water sources will actually keep BD zoospores longer, since there's less flowing water, that the spores can stay there longer and proliferate longer, and also due to the processes of eutrophication in these stagnant lentic waters, you would expect to see a higher microbial diversity, such as with BD as well. And so an alternative hypothesis to why lytic species have higher infection indices could be that maybe lentic species evolved to resist BD, or it could even be that that salamanders who live in these lentic environments could even have a more diverse microbiome to fight BD. Now, to put this into context, so studies have shown that lentic waters do indeed have higher microbial diversity than lytic waters. And that says that even though having a higher microbial diversity could be good for BD, but that means you also have potentially higher loads and higher diversity of other microorganisms as well. And so for these salamanders that live in these lentic waters, if they come into contact with these other microbiota, if you live in a more microbial diverse environment, chances are you could be picking up a microbe that helps you ward off infection. So an example of such a microbe is that of this bacterium called Xanthinobacterium lividum, or J. lividum for short. It's a bacterium that has been shown in lab trials to actually secrete antifungals on the skin of salamanders and actually helps them eradicate the BD infections. And so it's possible that this could be a reason why. Another alternative hypothesis is that stream habitats generally occur in cooler areas that are more optimal for BD growth. Again, BD is a fungus that prefers relatively cooler environments. Again, above 30 degrees Celsius, it pretty much dies. And so the thing about these stream habitats is a lot of times they're carrying water from upland sources, a lot of these upland mountain sources, and a lot of times you even get, a lot of water sources from, snowpack in the mountains. So it's oftentimes these streams carry cooler water into the environment versus lentic, stagnant waters where, it captures more thermal radiation. Therefore, average temperature is probably higher in these lentic environments versus these lotic environments. And therefore, it's possible that the cooler temperatures in the stream habitats make it more suitable for BD growth. Okay, then what about social behaviors that affect intraspecific contacts? So the thing is, these social behaviors may be important to BD susceptibility. However, it suffers from low data availability, such as territoriality presence, where there were only about 30-something species that had a trait value for it, or a lack of variation in data, such as egg guarding behavior, cannibalism in larvae, and communal nesting presence. And if you don't have enough variation in your data, you really just can't make a lot of meaningful analyses, come to meaningful conclusions regarding those traits. That said, in Plethodontidae, we see that yes values for communal nesting presence do confer a significantly higher average infection index, possibly since frequency of skin-skin contact is increased. And again, this is mostly due to Batrachosops. It can also be a function of the proportion of traits in the study that are related to intraspecific contact. Out of my 13 traits, only 4 of them, so about 31% of these traits related to social behaviors. And so having a study to measure the degree of social behavior could help improve our BRT results. What role do phylogenetics play? So the vast majority of our important traits have either moderate to high phylogenetic signal or low phylogenetic signal that was non-statistically supported, so it was not significant. And so the thing is, many salamander traits are actually evolutionary constrained. They're tied closely to the evolution of the salamanders themselves, meaning that certain groups, certain taxonomic groups of salamanders are very much defined by certain traits that they have. This doesn't necessarily mean that the traits don't contribute to infection likelihood. All it's saying is that we just can't say it's only trait-based. And so it's possible that resistance or susceptibility has a genetic component, despite these species occurring in a BD novel region and presumably not having the evolutionary time scale in order to develop resistance to it. And this would require further research. Looking at some of the at-risk species, so Batrachosops and Hydromantes, we see that 19 out of 22 Batrachosops species, so 86%, and 5 out of 5 Hydromantes species, so all 100% of Hydromantes species are at-risk species to BD infection. So for Batrachosops, this is especially concerning. They're a very non-vagile organism, which means they don't really displace much in their environment throughout their entire lives. So pretty much where they're hatched till the day they die, a lot of individuals rarely move beyond a couple dozen meters. So this can make them particularly vulnerable from being stuck in an area that is invaded by BD. And then BD has also been detected in a number of Batrachosops species in the field. And additionally, lab infection trials do show that they are indeed susceptible to the effects of chytridiomycosis from BD, meaning that, yes, they are definitely at risk should BD come in and sweep through the range. And then meanwhile, we see that for Hydromantes, this is a genus that lives in and around moist caves and fissures, which are generally optimal growing conditions for BD, even though there hasn't been too many studies looking at BD directly within these cave environments in the range that they live. So these salamanders are found in the Sierra Nevadas of California. However, studies in other cave systems around the world show that BD indeed can grow in these cave conditions, and that these caves are suitable habitats for BD growth, such as places like the Ozarks in Missouri, places such as Gabon in Africa, and even Italy has shown to have suitable BD growth in their cave systems. And lab infection trials show that the closely related genus Speleomantes in Italy is also highly susceptible to BD infections. And then looking at aquatic species at risk, we see that there was one species of Rhycotriton, one species of Tarica, and 3 species of Embystoma that are at-risk species to BD infection. Rhycotriton variegatus, one of those species, is a least concerned species. However, it occurs in coastal forests in the Pacific Northwest that are suitable for BD growth. And it is sympatric with BD harboring species such as Rana boylii, which is the foothill yellow-legged frog. And so if it lives in this environment where you have these BD harboring species and it comes into contact with these species, then you could potentially get skin-skin contact and skin-skin transmission of BD as a result. Meanwhile, for Tarica rivularis, it's also a least concerned species. However, it is endemic to a small area of northwestern California, which puts it at risk of extinction if a huge wave of BD invades its range. And so BD has been detected in Tarica rivularis. And additionally, lab infection studies of related Tarica species, such as Tarica turosa and Tarica granulosa, show that they are indeed susceptible to infection by BD. Continuing on, you see that there are 3 species of Embystoma that I deem as at risk: Embystoma leorae, altamirani, and rivuleri. And all 3 of these at-risk species are either endangered or critically endangered as defined by the IUCN, and they suffer from small specialized ranges, pollution, as well as urbanization and habitat loss. And so, especially pollution, this could exacerbate the effects of BD infection. And so in a previous study, again, looking at Rana boylii, the foothill yellow-legged frog, they found that when you expose Rana boylii to the pesticide carbaryl, that it actually reduces the skin peptide defense response against BD. So this could very well be the case in these Embystoma species where if they're exposed to pollution, it could exacerbate the effects of an impending BD invasion. BD additionally has been detected in Embystoma altamirani and leorae, suggesting that they may be vulnerable to disease-driven extinction. So looking at some of my study limitations, again, BD infection data is not available for all species. Infection index is purely model-based and has a geographic component. Infection susceptibility is not the same as infection intensity or morbidity/mortality. And so you might, if I had data for these things, I could potentially see and compare the trends between susceptibility and these other types of factors related to infection and see if the trends are the same. All these traits are qualitative based. Having more quantitatively measured traits would better help tease out patterns relating to infection susceptibility. And again, I would need more trait data to fill up my data set in order to have a more comprehensive study. There's a need to identify which genes, if any, confer resistance. And then for the study, I looked at each species as a unit. But what about subpopulation variations? There are some species of, say, Embystoma or Dicamtodon, the Pacific giant salamanders, for example, where some populations within a species retain the aquatic gill larval form throughout their entire adult lives, whereas there are other subpopulations that you see that they metamorphose from the aquatic gill larval form to the terrestrial lung form as adults. And seeing if there's any differences in infection index between these 2 types of populations could really help us get a better understanding of traits and their role in infection susceptibility. And then also, what about interspecific contact? So this whole time I talked about intraspecific contact, so contact within members of their own species, but what about contact with amphibians from other species? That's definitely a different aspect worth looking at in the future, especially, you could be looking at other amphibian species that are known carriers of BD and are known to be resistant to BD. So American bullfrogs are actually a good example of that. So expanding my study to look at these BD carrier species will definitely help strengthen the study. In conclusion, we see that habitat preference affects infection susceptibility the most. Non-aquatic species have the highest infection susceptibility when they're both terrestrial and subterranean, and lowest when arboreal. We see that aquatic species have the highest infection susceptibility when they're lotic, and the lowest when they're lentic. We see that Batrachoceps and Hydromantes, which are both plethodontids, are particularly at-risk species of BD infection. We see that social behaviors may play a key role, but more data is needed. Traits generally have phylogenetic signal, meaning infection susceptibility may also be genetically based. And ultimately, we're going to need more trait data, especially quantitative trait data, more infection data, genetic data, and subpopulation data to improve my BRT analysis. For my references, I want to say thank you to students over at San Francisco State and at the Museum of Urban Zoology, the MVZ, at the SF State. I just wanted to say thank you for helping compile the list of traits that I use in my data set, and to the students at the MVZ, thank you for helping create the range maps for the different species that I use in my analysis. Without the efforts of all these students, I would not have been able to get the data that I needed for my study. Additionally, I want to say thank you to, of course, my committee, Vance, Michelle, and Andy, for helping me with the guidance and support throughout my project. Additionally, I want to thank Hassan Soliman for helping me with compiling some of my BD occurrence data sets. And additionally, I want to thank Josh Banta and Tiffany Yap for help with species distribution modeling. And with that, that is my presentation, and I will take questions. Thank you. Right, so yeah, I'll gladly take any questions from the audience if anyone's curious.
Hi, Gordon. Is anyone else trying to ask a question, or should I just jump in right now?
You can just jump in. You can just jump in.
Okay, great presentation, by the way. Great defense. Loved it. Terrific. Congratulations. So
Thank you.
I just have one question for you, and this, and it's how are you doing the validation for the models? And also, I've worked a bit with the Pyron data set for the phylogenetics. And I know that for every species, it's not like every site that gets like sequenced. It's like it's pretty different. It's pretty variable across multiple species. So how are you accounting that in your modeling? So
like you're saying that for the Pyron paper that he used like sequences from different individuals within the species that may not represent the entire species?
So the sites that are being sequenced for Pyron to build that phylogeny, it's not like the same site. Does that make sense on the genome?
Oh,
so like,
okay.
Yeah. For 15 species that I was trying to work on, it was like very, very cool. So like, yeah.
So like you're saying like maybe like for one species, you use like the 16S gene versus like a different gene for another species?
Yeah, some species have like 3 to 4, and some species only have.
Yeah, no, that's definitely a really good question. Unfortunately, I was not able to account for that. I mean, I just had to take in good faith that the data presented in the Yetson Pyron paper was trustworthy. And I mean, I'll put it this way. Yes, obviously, you would want to look at the same sequence, the same sites for all the species when making such a tree. I will say that in terms of regarding my study in particular, yes, it's important to have that, but I'm more interested in looking at general trends and phylogenetic relatedness, and maybe not so much like the deeper intricacies of phylogenetic relatedness, such that even though it's not perfect, I'd still say that in terms of the overall picture and just getting a sense of, is there any measure of phylogenetic signal? I would like to say that that suffices for my paper. But yes, definitely having the same sites for all the species is definitely important. And then in terms of validating my model, you mean for like my Maxent model, Hassan?
Yeah, for Maxent, I know there's like Jack built in. Like you have the RBT too, right?
What's up?
The regression tree.
Yeah, sorry. No, I have the regression tree for the BRT analysis. Yeah.
Oh, yes, that analysis. Sorry.
Oh, sorry. You're wondering, how I validated the model. So, so in order to do that, I actually would run each time I had the different taxonomic groupings. I actually ran the model a number of different times using different parameters for like tree complexity, learning rate. For example, I basically would just kind of play around with these different settings. So like, maybe one instance I would set the tree complexity to 10. Another I would set the tree complexity to 5. And then I would also just, set the learning rate to different values just to see what the output was. And if the outputs were generally the same, despite me shifting the parameters, then I could say that, the trend holds pretty strongly for this data set that I have, and therefore the model fit is pretty good. And also, even when I did find, so ultimately I would want to get model settings that minimize predictive deviance, right? And so even when I narrowed in the certain parameters, I would still run those settings a number of times just to make sure that the minimum predictive deviance was the same, and I would also compare and make sure that the RI values were the same. So yes, this is a stochastic process, these BRT analysis. So every time you run it, you are going to get slightly different values. But the most important thing is just looking at what is the degree of difference in values each time you run the BRT. And as long as it, came out within like, maybe a couple points, a couple like RI values difference, nothing too big. And as long as, the hierarchy of the important traits was still there, then I figured that that was a good model fit.
Yeah, awesome. Thanks so much, Gordon. Great work.
Thank you.
Thanks.
Anyone else?
Hey, Gordon, this is Vance. So I wanted to ask, first of all, great job. You really pulled it together there. And I just wanted to say, so you had 350 species in your analyses out of 352 that occur there, right?
Right.
Yeah. So first of all, it's great to be so complete where you're able to, it's really hard to do a study where you include every single species within a geographic range like that. So it's really good. But I guess one question I had on my mind as you were describing the results that I didn't see is out of those 350, like each one has been assessed in terms of its conservation status, correct?
Right.
And how do your, first of all, what are the general status? Are they in as bad shape as frogs are or not? And then secondly, is there any relationship between the status, conservation status, and what you're predicting from your model?
Not really, to be honest. I mean, I don't have the exact list in front of me right now of all the IUCN like the status levels, but there were a lot of species that were least concerned, as you can see just from a number of the species that I included that were at-risk species. And a number of the Batricus species, yes, some of them are quite like vulnerable or endangered, but there's also a number of other Batricus species that were least concerned species. So I would say in terms of how well does it match up with conservation status, probably not as well as I would think.
I guess I would just ask you to sort of expand on that. Like, it's not like they need to match, but if they don't match, why, what are your thoughts on that?
Right. So my reasoning is either these species need to be assessed more and take more into consideration the threat of disease. So when you look at the IUCN statuses, when they mention like, how they evaluate it, such as like the different threats, they'll mention like, habitat destruction, pollution, urbanization, things like that. And they'll like mention disease, but it's never in deep detail. So I would challenge that the folks at IUCN, when assessing, these species, if they do a reassessment, to have, more infection data to look at to help them make more meaningful conclusions as to their, true conservation status, shall we say. And additionally, even though these species are at risk, I mean, they could still be a least concerned species just by virtue of maybe the fungus has not quite yet invaded their particular range as fully as compared to like other species. So, so in order to be an at-risk species, in order to actually die off from the species, you need two things, right? First, you need to have the traits that help you maximize your your contact with BD, either through water or skin-skin contact. And secondly, you would need to actually know that the BD is actually there presently invading in that range. So if you put those two things together, that's when you actually see huge die-offs related to BD. And as long as you only have like one out of those two criteria, then you're not necessarily going to see die-offs related to BD, and as a result, a conservation status that reflects that.
By the way, Helen has her hand raised.
Oh, hi, Helen. Any second now. All right, haven't heard.
That may be a mistake. I don't know.
Oh, okay.
Could I jump in?
Yeah,
sure.
Yeah, I have a question too. Go ahead.
Oh,
Yeah,
no, I'll wait. I'll put my hand.
Yeah.
Hey, Gordon, I have a question. This
Yeah.
fella. From your map of BD positive versus and BD negative points, it looked like you had clusters of points in the Pacific Northwest and sort of central coastal California, south of the Bay, that were negative. Is that right? And can you kind of
Yeah, there was a cluster. There's a cluster of samples in like California, then another cluster, southern Mexico and Central America.
And also up in the Pacific Northwest, it looks like most of those points were BD negative. Was I looking
Most
at
of
that
them
right?
were, yeah, negative. Yeah, that's just, I mean, I guess a limitation of the data sets that I had available to me. I guess, I mean, yeah, definitely, sampling bias is definitely an issue. And so to really further strengthen such a study, yeah, I would want to get more data sets, or, if there's not enough data in that area, then send more people to go out and sample those areas to actually say, whether or not BD occurs in that area. Yeah.
So you think it's a sampling issue and not like a trait issue with the species that are associated that occur there?
Possibly, yeah. I mean, yeah, and I get what you mean. I mean, I don't see why the Pacific Northwest wouldn't be a good place for BD. It makes sense. The conditions are great. It's cool. It's very wet. So presumably those are good conditions for BD. And yeah, I mean, so yeah, we could definitely benefit from more sampling. And, I definitely tried to account for that fact that, that area wasn't quite captured as well as it would have liked in terms of sampling. And I was hoping, that's kind of what the hope is in creating a model that minimizes overfitting, is to more accurately capture potential areas with high BD suitability. And so even though in the map, BD suitability could have been higher in that area, I still did see levels of, at least moderate suitability. So in the Pacific Northwest. So it's not perfect, but I did my best to account for that.
Okay, all right, thanks.
Yeah. All right, anyone else?
Yeah.
Hi, Gordon.
Oh,
Hey, Ellen.
Go ahead.
no, go ahead. I'll wait.
Hey, Ellen.
Hi, Gordon. That was awesome. That was really fun to listen to. So you kind of hinted it at in your closing remarks in terms of genes that may or may not have an effect on susceptibility. And I was really interested in your kind of searches you came across any data sets on like the innate immune system and whether or not you think that, I mean, I'm sure probably the data is lacking, but if like complement cascades or anything differs between these species that might later on when the data exists be added.
Right. I mean, I admit I did not look as closely as I could have into that. I mean, you probably know more about that aspect than I do. I do know that, in a lot of amphibian species that are resistant, that they show different levels of like gene expression, especially with regard to the major histocompatibility index, or the MHC. So I would imagine that for species that are indeed, have levels of resistance in my area, that some of them would have genes to express, the MHC. I mean, that's really all I can say, unfortunately. Yeah, I think we would just need more data, and that definitely sounds valid. Yeah.
No, that's great. Thanks.
Yeah. Anyone else from the audience?
Did you want to go, Andy?
No, you go ahead, Michelle. I'll go next.
Okay. Thanks, Gordon. Saw a lot of great improvements. So I just wanted to ask a couple of questions, both general and specific. One of them is, so you focus really on BD here and salamanders, but the big chytrid fungus issue that, of course, that you alluded to is the B-sal, or you talked about that at the beginning. So I'm just curious, what are your thoughts about your particular methods and your analysis, and whether or not you think either the results, the methods, the means, if any of that is transferable to discussions about B-sal and vulnerable traits?
I mean, I don't know the exact physiology of B-sal. Michelle, maybe you could help me with this. Do you know, like, how similar is the physiology between both species?
You mean about the actual fungus? They're pretty similar.
Yeah.
They're related.
The actual
fungus.
Yeah. Okay.
Yeah.
Well, if they're pretty,
but there's a lot that's not known too. But yes, in general, they have similar.
Right. Well, assuming their physiology is similar, assuming their biology is similar in terms of, transmission, in terms of, their habitat preferences, I would say that, the results from my model could probably transfer quite well over to B-sal. I would hold off on making an actual judgment about B-sal until we understood more about its biology, but I definitely think it's a good starting point. And, you know, in conjunction with your 2015 paper with Tiffany and Vance modeling, you know, the averting a North American biodiversity crisis, looking at BSAL, where it could potentially invade in North America. I think, yeah, I think it's definitely, I definitely think that what I see could carry over, yeah, really well to BSAL, just by virtue of them being, you know, very related species with, as of that we know, similar physiologies. So I think the trends could apply over.
Mm-hmm. And so similarly, how well, but not about B-cell specifically, but more about BD. So you talked a lot about, obviously, sort of the phylogenetic component, since your data set was so heavily weighted towards plethodontids. And that obviously that's because, you know, you're working in the salamander hotspot of the world. What about your thoughts on if you were to transfer your analysis to the salamandrid hotspots of the world, or the other, and the Bistomatidae, or, you know, the other families that are also speciose, they're just not super speciose in your study area?
Well, regarding embystomatids, I don't know how much we could say about that, because a lot of embystomatid diversity, besides the Western United States and Mexico, the rest of it is in Eastern United States. And since I already previously mentioned that the Eastern United States is one of those areas where BD is an endemic pathogen and we don't see a lot of related die-offs, that unfortunately, I mean, even if you get, you know, results out of a study looking at Eastern US embystomatids, that just by virtue of them not being known to have a lot of BD-related die-offs in that area, I don't know how strong of a claim you can make about salamanders in those regions.
Well, we haven't, just to be clear, we haven't had a lot of salamander die-offs in Western North America either.
That's, well,
I mean, you know, right?
True. I mean, I guess in that case, you would want to more closely look at, first do some lab infection trials of like the Eastern United States embystomatids, see which ones of them are actually susceptible to chytridiomycosis. And then from there, once we figured out which of those Eastern US species might actually be susceptible, then we could actually run a BRT analysis. And I would expect that probably the results would be fairly similar to what I'm seeing with my Western US embystomatids. And regarding salamandrids in other parts of the world, I know there's a lot of salamandrid diversity, say in like Asia and Europe. Asia, there's not nearly as many BD-related die-offs. So, and I do know that a lot of species there are, you know, show some level of resistance. So I don't know how well my model would transfer to these resistant salamandrid species. In terms of the species in like Europe and North Africa, I'm trying to remember, I think they might be slightly more vulnerable to BD, well, especially B-Sal for sure. I think in those cases, yeah, you could expect to see that the results would come out that, yeah, potentially species that are more, let's see. Well, in my study, I found that when it came to Tarica, that on average, that the higher infection index, or for salamandrids, that species that were territorial on average had a higher infection index than those that weren't. However, due to the spread of values, I couldn't really say too much about it. I think that if we actually went in and recorded the territoriality status of the different salamandrids and those other parts of the world, that, I mean, I would think that, you know, depending how you characterize territoriality, you know, some people would say that territoriality results, you know, depending on the species, territoriality could result in increased contact because of like fighting each other, or sometimes in actually more isolation between individuals just because they have larger home ranges. So I think in that case, you would actually want to further divide territoriality, because for my definition of territoriality, it's fairly vague, to be honest. I think your study looking at salamandrids could benefit from a more specific definition of territoriality, and maybe even having like a couple, breaking up territoriality into a couple of different traits. And then when we look at that, we could see that maybe potentially species of salamandrids that engage in, you know, direct like bodily contact as a result of fighting each other probably have a higher infection index, whereas these that are territorial, but are territorial with respect to maintaining larger home ranges and isolating themselves from other individuals, would presumably have lower infection indices.
Yeah. Yeah, interesting. And then lastly, I wanted to ask, you mentioned in your sort of study limitations, but I also sort of took it as like a, future perspective studies. You mentioned something about quantitative traits that would be desirable to see. So could you give some examples of that? Maybe you all kind of already did in some ways, but
yeah, I mean,
I'd like to hear more about that.
The example that I showed in my my defense just now was that of the Raleigh-Alford 2007 paper, where they actually measured the frequency of contact with streams and frequency of contact, like direct skin-skin contact. So I think putting actual numbers to these qualitative traits would improve things very greatly. In terms of the other traits, it doesn't necessarily have to be streams, but just like general contact with water whatsoever, or when looking at these non-aquatic plethodontids, measuring what amount of time throughout like a certain season or even throughout the day do they spend underground versus on the ground versus in the trees, depending on the species that you're talking about. So actually putting a percent frequency to that amount of contact would really help. Regarding other traits, I'm trying to think of, if you look at territoriality, presence, maybe like amount of time they also like just come into contact with other individuals. If we're looking at cannibalism in larvae, like looking on on average for the cannibal ecomorphs that are present within a certain species, in an average environment for those cannibal ecomorphs, how many other larval young would those cannibal ecomorphs consume? And having like an average number could also help inform analyses, rather than just saying, oh yeah, we know that the species has cannibal ecomorphs versus the species doesn't have cannibal ecomorphs.
Right, yeah.
Yeah.
Okay. Thanks for your thoughts on that.
Andy?
Hi, great job, Gordon.
Thanks, Andy.
I've got two main questions.
Okay.
The first is with respect to the plethodontids species that had a really high infection index that you found. I mean, a lot of them were in the genus Batrachoceps.
Right.
We actually have a fair amount of data now on for those species at least, showing that the prevalence is kicking around 15%, even though it's been widespread for over 3 or 4 decades. So why do you think there's a disconnect there? What do you think it might be that's keeping BD at a moderate level? What is it about the life history, ecology of these different species?
Right. So I think, well, you do have to keep in mind that when I'm doing my habitat suitability models, this is just a model of suitability, right? This doesn't, habitat suitability models model suitability, but they don't model actual presence of the species. So that's definitely one thing to keep in mind. So if I'm saying that the species has an infection index of 0.5, all it's saying is that throughout its entire range, it has it occurs in 50% suitable BD habitat, or it's 50% likely to come into contact with BD in its range as the species overall. So this is just modeling suitability, which is a different aspect from the actual field data. Of course, it would be nice if the two things matched up closely in tandem. I think if we do see any discrepancies, it could also be a result of the fact that, maybe BD has not quite yet had a chance to invade the entire range. I think that's another possibility. And so all it's saying is, this is just saying that these species are 50% at risk if BD does in fact invade its entire range. So maybe, the entire range has not been fully invaded yet. Or alternatively, if you remember from the Weinstein study that I mentioned early on, potentially that, even though we're talking about habitat suitability of the fungus in terms of its overall range, you still want to account for microclimate, microhabitat differences, and that could also have a part to play in BD, right? So ultimately, even though this is looking at general trends in terms of large ranges of area, really what's most important is looking at the minute scale, looking at the microhabitat usage of BD, as well as the microhabitat usage of these Batrachoceps species. And so I think if we had a better understanding of these Batrachoceps species, it could stand a reason that maybe, some populations of Batrachoceps occur in slightly more arid microclimates. Like even though if they live in a high BD suitability area, they could potentially position themselves in a part in the environment that puts them less at risk of coming into contact with the fungal pathogen, just because they purposely or subconsciously avoid like wetter patches of soil, for example.
Yeah, they burrow pretty deep into the soil as well. They have very limited ranges. Yeah, thanks. That's a great answer. My other question was around behavior. You spoke at length in the discussion around communal nesting, and that's a very intuitive and straightforward behavior that that would be expected to correlate with your high infection indices. Now, egg guarding is less intuitive.
It is less intuitive.
Yeah, and it could, of course, be some. So I guess I have two parts to this question. If it is actually a direct causative factor, which I know you haven't proven, and there's limited data, what would be the mechanism? And if it's not, then what might be some other factors that would be correlated with the propensity of certain species to egg guard that might also correlate with an ecology that would be suitable for BD?
Right, so let's see. So when looking at egg guarding behavior, if that is indeed the case. Sorry, can you repeat your first question again, Andy?
Yeah, sorry. I'm just thinking about how BD could be spread more easily through egg guarding.
Through egg guarding.
Yeah, it just doesn't seem very intuitive.
It doesn't. Well, I mean, I know from previous studies they've shown that species that do egg guard could actually help decrease infections and disease on these eggs, because with brooding you can help you can take care of the eggs, you prevent like predators or other like pathogens from spreading, and you could also like clean the eggs physically to yourselves of the infection. So I think that's one way that the fungus could that BD could be contained and spread, is if these species that do egg guard do indeed take care of their eggs more and prevent them from being infected. If that weren't the case, I would expect that, I'm trying to think, that it could be just more of a function of other more general life history traits in those species of salamanders, and maybe traits that are more pertinent to habitat preference. So if it's not because of egg guarding behavior, I would just think that probably there's more just a correlation with respect to preference for like subterranean versus terrestrial versus boreal habitats. And I mean, just from the first pass of my study, we see that ultimately these traits are the most important. It's not to say that social behaviors aren't important, but it potentially suggests that habitat-based traits that are relevant to water contact probably have a more important factor to play in terms of BD infection susceptibility versus these social behaviors. So I would say, even if it's not the case, it ultimately comes down to just from what I've seen in my study, that these habitat-based traits matter significantly more than the social behaviors.
So just to sum up what I think you said, which makes sense, is because you would expect egg guarding for many species, it's been shown with like the four-toed salamanders that they can actually deposit beneficial microbes that will reduce BD. It could just be an autocorrelated situation, and that environments that are pathogen rich or likely to sustain fungal disease would select for maternal care of eggs, and therefore be correlated with the presence of BD. Sort of
that's quite possible, right? Yeah, the environment itself could definitely have a part to play in it. Yeah.
Thanks. Yeah.
Great, thanks, Andy. Questions from anyone else? All right, Vance.
Raise my hand. Hey, Gordon. So yeah, I just had another question, which was, one of the two questions. One of the things you found was that the type of habitat they were in mattered, right? So if they're arboreal, they're less likely to be at risk. If they're subterranean or terrestrial, they're more likely to be at risk. However, most of your species are subterranean or terrestrial, I believe, right? So how do you, the sample sizes basically amongst those traits in your species are not equal. So when you look for a pattern, if you have a lot of replicates of these terrestrial ones, for example, they're probably more likely to overlap with BD at
Right.
all. So how do you know if that's a real result or just a result of your sampling and your
Have the
methodology?
sample size. No, that's definitely true. I mean, ideally, you would want to have at least good sample sizes for each trait value. So, like, generally, statisticians say that having a sample size of 10 is usually good. So, a lot of these trait values, even though, yeah, a lot of them were like terrestrial / subterranean, but even for the other trait values, except for like, species that were all three, arboreal, terrestrial, subterranean, but for the other traits, a lot of them did have a number of, a good sample size of species. So I would say that, even if the sample size is not as large as the terrestrial / subterranean species, it's still enough that we can still see general trends. And I think, yeah, to answer that question, like how would I account for that? I think, yeah, I mean, have another study where you focus on, those specific species that do have just those trait values. So maybe instead of focusing on species that are both terrestrial and subterranean, then maybe focus your efforts, look at the species that are not terrestrial and subterranean. Look at the species that are just arboreal or just terrestrial or just subterranean or a combination of these. And so, I mean, I know I didn't do this in my study, but I think to answer your question, in order to account for that, I would, want to make additional studies and maybe a future paper looking at just those species. Well, first I would have to establish that terrestrial subterranean species have the highest infection index. Once we have that established, then we can say, okay, so for the rest of the species, what trends do we see in those other species instead? And then from there, we can go on, look at those species more closely in detail.
Okay, yeah, I'd agree. That's a good way to handle it.
Yeah.
How does the other way to handle it is to make comparisons, right? So how do these specific results relate to what we know in a much better studied group of amphibians in terms of BD, which is frogs? How do they relate?
Yeah, well, I mean, if you remember, I talked about the study in Cameroon where they looked at the frog species there, and they found that frog species that were arboreal tended to be at less risk, had a less risk of BD on their skin. So when they swabbed these different amphibian species, they swabbed these different frogs in Cameroon, what they found was that, yeah, the frogs that were arboreal generally had a lower prevalence of BD than the species that were lower to the ground and utilized streams and ponds more. So, I mean, that's just, one study that I can think of right now, but I'm sure there are other examples in literature that suggest, again, that being closer to these sources of water can definitely help increase the likelihood of BD contact. And there's also the effect of temperature fluctuations with respect to ground temperature versus air temperature. To my knowledge, I don't think people have looked that closely into that particular aspect. But I think, yeah, in the future, it'd be really cool to have a study to look at, air versus ground temperature fluctuations in an environment where BD is known to be present, look at species that are found in those arboreal habitats versus those ground habitats and see if there are differences. And I mean, if I'm just going to hedge my bets, I have a feeling that we probably would see similar trends to what my results found. Okay, thanks, Vance.
Great. Anybody else have questions? Yeah. Right. Well, great. Well, thank you so much, Gordon, and thank you everyone for coming today. And really look forward to seeing where this goes in the future, Gordon, and congratulations on giving a great talk.
Thank you so much. Thank you, everyone. Thank you, everyone, for coming. And I'll talk to you guys soon.
Thanks. Bye, Gordon.
Yeah, thanks, Gordon. Congratulations.
Thank you. See you all soon.
See you soon.